peoples from the north side of the river and the Jaguar peoples from the south side of the river. We acknowledge also the traditional owners of the lands which you will occupy today, and pay our respects to elders past, present, and emerging. In talking about reforms to food production, it's probably worth recognising that for tens of thousands of years, the Indigenous peoples of Australia practiced more or less sustainable land and food cultivation techniques, some of which we can still learn from today. So let's talk about food production now. Global population is on track to reach almost 10 billion by 2050, and demand for meat will rise with this population increase. But current meat production techniques are environmentally unsustainable, economically wasteful, wildly dangerous from a public health standpoint, and if we consider the experience of the animals we raise in factory farms for even a moment, pretty monstrously unethical. So if we want to end all this, there are two broad ways we can go about it. Firstly, we could try to get people to change their own consumption behavior voluntarily through moral persuasion and argument. But some of us have been trying that for a while, and it's slow going, to say the least. So alternatively, we can aim to make current farming practices technologically obsolete so that no one even considers animal farming as an option. And that's the alternative that we're here to talk about today. To achieve this goal, we need advances in science. In particular, we need to figure out how to produce quality alternative meats that mimic the taste, texture, and nutritional quality of animal meats. We need to figure out how to do this at scale and at price parity with animal meat. And we need consumers to accept the end product. Tonight, you'll be hearing from researchers who are working on the cutting edge of these very problems. This event is brought to you by the EAUQ Effective Altruism Club. Effective altruism, for anyone who doesn't really know, is all about using evidence and reason to try to figure out the best ways to do good in the world from an impartial perspective, and then doing it. So if that sounds broadly good, then I do encourage everyone to click the link in the chat below and follow the EAUQ Effective Altruism Club on Facebook. It's a great space to discuss these really important things and for more events like this. And we're nice people too, I think. Also in the chat, you'll find a link to Slido, which is a website that we'll be using to ask questions throughout these talks, and where you can vote on which questions from others you'd like to see answered. Please do use that link instead of the chat itself to submit questions. It's really easy to miss things on Zoom. We don't want to miss questions from anyone. So yeah, Slido in the chat below is your place to go for questions.
Can I just jump in and add, if you could also include in your question who it's directed to, if you want a specific person to answer it.
Great point. And we have 4 people for you to ask questions for. Well, 4 people speaking. Ruth Purcell has experience with alternative meats in both academia and in industry, having worked as a researcher at ANU and as a synthetic biologist for Nourish Ingredients, a food tech company which creates animal-free fats and oils for alternative protein products. Ruth now works helping decrease pandemic risk at the Peter Doherty Institute for Infections and Immunity, which is another global priority we talk a lot about in effective altruism, by the way. And she volunteers at Cellular Agriculture Australia, a non-profit devoted to improving research, collaboration, and innovation across the cellular agriculture sector in Australia. Ruth will be talking about the state of the alternative meat industry in Australia, with a particular emphasis on precision fermentation techniques, which show some real promise. Like Ruth, EAUQ's own Professor Jason Stokes has diverse research experience, both in and out of academia, having worked as a research scientist for Unilever's corporate research laboratory in the UK for 10 years. They're the company that makes everything from like chocolate to like deodorant. Working from the School of Chemical Engineering at EAUQ, he has a pretty broad research focus and is currently partnering with the Boston food tech startup Motif Foodworks on investigating how to develop new plant-based alternative meats that realistically mimic the taste and texture of animal meat and live up to consumer expectations. Dr. Felix Septianto knows a lot about consumer expectations. He researches the influence of feelings and emotions on consumer decision-making. Felix is one of the world's most active marketing scholars and has recently received an Australian Research Council grant to investigate the emotional factors affecting consumer acceptance of alternative meat. This is one of the biggest challenges the industry faces moving forward. If consumers don't accept alternative meats, then everything else we're talking about in terms of techniques is sort of null and void. So I'm really keen to hear from Felix about this. And finally, Dr. Mark Allenby leads the Biomimetic Systems Engineering Lab here at EAUQ, which brings together a range of bioengineering techniques to study and solve biological and medical problems. Many of these techniques are also applicable in the construction of cell-grown meats, and collaboration between medical tissue engineering and tissue engineering for alternative meats has been fruitful in the past and promises to be so again in the future. Mark will be talking about some of the scientific nitty-gritties of tissue engineering cultivated meats, and we're lucky to be hearing from a real expert in this area. Just so you all know, Mark and Felix are both off being busy cutting-edge researchers tonight, so we'll be hearing from them in a pre-recorded format. Despite Ruth and Jason also being cutting-edge scientists, however, they've found time to join us live, which we really appreciate. And finally, before I shut up and hand things over to Ruth to start us off, I just want to say a big, big thank you to Philippa for organising this event. I am just the host tonight. Literally all I've done to prepare for this event is like shave. Philippa's been the one sending emails, organising dates, and curating this awesome lineup of speakers. And Harry Lee Jones has also been a real help behind the scenes as well. So for anything valuable you get out of this event, thank Philippa, thank Harry. I just shave. With that said, I'll now pass you over to our first speaker tonight, Ruth Purcell.
Just going to share my screen.
We can see it, Ruth.
Yep.
Awesome.
Great. Thanks, guys. Thanks for the introduction. So I'm going to be talking about cellular agriculture, giving you a broad overview of what it is and what the industry looks like in Australia. And I'll just start by saying that, I volunteer at the registered charity, Cellular Agriculture Australia. Oh no. Oh dear. Sorry. Okay. All right, we're all good. Sorry about that. Okay, so talking to an effective altruism crowd, I figured it's pretty safe to assume that a lot of you spend a good amount of time thinking about the major challenges humanity faces. I know I certainly do. And for me, there are really four big standout ones. Climate change and biodiversity loss, food insecurity, antimicrobial resistance, and increased animal to human disease transmission. So applying the effective altruism philosophy of doing the most good we possibly can with the limited resources, time, and energy we have available to us, the first step is, of course, to step back and figure out what unites these four challenges so that we can address them at their root cause. In this instance, it's industrialized animal agriculture. Of course, the goal of tightly packing animals into small spaces wasn't to cause deforestation, land degradation, and increased disease spread. It was to increase the efficiency of food production, which we did to some degree, but in all fairness, animal agriculture is not that efficient. For example, it takes 8 calories of feed into a chicken to produce just 1 calorie of food for human consumption. For beef, it's even worse. It takes 33 energy calories into the cow to get 1 calorie of beef out. At a 3% conversion rate, I don't think we're doing too well in terms of efficiency. And then there's the matter of land use. Of the 21% of the Earth's habitable land, half of it is used for agriculture. And of that, 77% is taken up by livestock to produce just 1/3 of the world's protein supply. So with the human population rapidly increasing, and with there being not much more land to expand into, how on earth are we going to feed almost 10 billion people by 2050 without absolutely annihilating our planet? Well, the answer is simple, right? Let's all go vegan. Yeah, that's not working too well. In fact, we're trending in the opposite direction. meat and animal products are really, really delicious, and behavior change is hard. So as the middle class grows, so does meat consumption. We've done pretty much all we can to change the demand for animal products, and it's not working. So it's time to change the supply. And this is where cellular agriculture comes in. Cell ag is the production of animal products directly from cells instead of from live animals. So instead of growing a whole cow with hooves and horns and sentience, we can just grow the bits of meat that we want to eat. This is not to be confused with plant-based meat, which I'll touch on in a little while. So how does cellular agriculture work? First, we take a small biopsy from an animal which contains stem cells. These stem cells are then exposed to proteins called growth factors, which induce proliferation so that we get an increase in the number of cells and we let them multiply. Once we have enough cells to work with, we can expose them to different growth factors which induce differentiation so that these cells change into all the different cell types we see in a piece of meat. So fat, connective tissue, muscle cells. So that's all well and good, and the technology is really cool and exciting, but how efficient is it really? Well, compared to 3% and 13% for beef and chicken respectively, I'd say with an 80% energy conversion rate, we're doing pretty well. So now that you have an idea of what most people are talking about when they refer to cellular agriculture, so cell-based meats or cultivated meat, let's talk about a different branch of cellular agriculture which gets a little bit less attention, and that's precision fermentation. So precision fermentation is the use of genetically engineered microbes to produce a useful product, such as proteins or lipids or flavor molecules. And an example of this is from the company Eden Brew in Australia. They're working on creating animal-free milk. So without using a cow, they're able to make dairy proteins which perform and taste, or soon will perform and taste, in the same way that milk does. And in fact, their goal is to get milk that is so functionally close to real dairy, you won't notice the difference in your coffee. Again, how does this work? It's not that different to the way we produced cultivated meat, in that we take our desired cells and grow them under optimal conditions in a bioreactor. So precision fermentation is also referred to as acellular agriculture, or more commonly, recombinant DNA technology. And I'm sure many of you are now thinking, oh, I've heard of this before. And for all the scientists out there, biologists, you've probably done this yourself in the lab. It's pretty standard molecular biology techniques. Precision fermentation is fairly simple in that all you need to do is identify your target gene, which will produce your desired protein or lipid, insert this gene into a microbe, such as yeast or bacteria, grow these microbes under optimal conditions with the right pH, temperature, oxygenation, nutrients. And what that'll allow you to do is then grow enough of your cells to an adequate quantity and then extract your target protein or lipid, which can then be incorporated in an animal-free food downstream. And to me, the most exciting part about precision fermentation is that this novel concept isn't all that novel. In fact, precision fermentation, or recombinant DNA technology, is how we've been making insulin for diabetics, rennet for cheese making, for decades. The technology is pretty well established, it's well understood, the platforms exist, and it's fairly well regulated. And for this reason, precision fermentation products will probably be the first cell ag foods that we see on Australian shelves. Because it's really well understood, it's much easier to incorporate into the foods that already exist. And not only the fact that it's well understood and well regulated, but Australia also has a thriving plant-based industry, and this too will support the introduction of precision fermentation-based foods before we see cell-based meats on the market. So V2 burgers and V2 plant-based meats are examples of Australian plant-based food. And I'm sure many of you have actually tried them. If you were anything like me, you're probably quite disappointed when you bit into a plant-based meat alternative. At the moment, the taste and texture just does not live up to what we expect from something trying to replicate meat. It simply isn't the same as an animal product at the moment. But what this does mean is that these plant-based foods are the perfect canvas for the products of precision fermentation. It only takes a small amount of lipid or protein to completely change the texture and sensory and functional profiles of these foods. It's actually quite incredible the tiny amount of lipid can do to an otherwise completely bland product. Lipids are able to carry so much flavor and completely transform the plant-based meat. In fact, it's products of precision fermentation that I think will take plant-based meats from the compromise that they currently are to being in direct competition with animal-based products. The real difference between this and that is fat. Using the right fats in the right proportions can completely transform the consumer experience of these plant-based meats. So these examples from Juicy Marble, the real difference here is the fat. In plant-based products such as the V2 burger, these white flecks are bits of coconut fat. And coconut fat does not perform in the same way as animal fats. But when you use microbes that have been transformed with the genes from the animals of interest and you're producing the correct animal fats, you get a completely different sensory profile, which is actually convincing. So this leads me to the wonderful Australian precision fermentation company I was lucky enough to work with, Nourish Ingredients. And Nourish Ingredients is using yeast to produce animal-free lipids for alternative proteins. So this sounds like something that's interesting to you and something you'd like to get involved in. Australia is the perfect place to be. As you can see, in the cellular agriculture ecosystem in Australia, many companies are using precision fermentation. These three are working on proteins, while Nourish Ingredients is of course working on fats. Then we have companies that are working on whole cut meats, as well as the auxiliary products required to grow these cells into whole cuts, such as the feed or the culture media and scaffolds to support the cell. And because Australia is not boring, we have a really diverse range of products that's even including cultivated breast milk. And for those of you looking to stay in academia, we have a number of research labs which is growing as we speak, in which you can get involved in cellular agriculture research. So how do you transition into the industry and how can you get involved? Cellular Agriculture Australia has put together a really effective pathways tool in which you can look up all the different majors required in the field, as well as the knowledge gaps and problems to solve in cellular agriculture. And the one thing I really want to highlight, especially given that the audience here is so diverse, is that this is not just science-based. It's an all hands on deck problem. We need everyone, from marketing and marketing specialists, lawyers, accountants, business strategists. It's not just scientists who are needed. There's a place for everyone in cellular agriculture. So on that note, I would just like to leave you with 3 ways that you can stay up to date with the cellular agriculture space in Australia. You can sign up to our newsletter, follow our social media channels, and join our Slack page, which is a vibrant community of about 150 people, not just in Australia, but also from a few other countries, discussing cellular agriculture and right at the leading edge of the technology. There are heaps of job opportunities available, as well as other general interesting discussion points. And you can even meet the team behind our work. So with that, I'd just like to thank everyone at the Cell Ag Australia team, and I'll leave you with the website in case you want to go and follow up and get involved in the work we do. Thank you.
Thank you, Ruth. That was great. I had no idea personally that we actually just had such a vibrant and growing industry in Australia. You hear a lot about food tech companies in the US, which are really pushing ahead with this kind of stuff, but it's awesome to see that we've got the same kind of thing happening here.
Yeah, I think that was Bianca's big gripe when she founded Cellular Agriculture Australia. Just the fact that so much attention was poured into the States, and the Australian cell ag ecosystem was just really underrepresented, not well connected.
Yeah, no, it's awesome to see that people are doing good work to make it come together. And for everyone observing, Harry's just put the link to the Cellular Agriculture website in the chat as well. So feel free to click away for your heart's content. Fantastic. I would ask for a silent round of applause, but cameras are also off, so I don't think we'll receive much there. But from me, thank you. So that takes us into our second talk, and we'll move quickly into it. Jason Stokes on plant-based meat alternatives and mimicking taste and texture with advances there. We already touched on that a bit in Ruth's talk with talk about different precision fermented lipids that could be augmented into plant-based technology. There are questions for Ruth. We have questions for Ruth. Okay, we'll sort questions first, if we can, and then we'll come back to you, Jason. Sorry for pulling ahead there, folks. So first question, bit of a lighthearted one, but just personally, Ruth, what's your favorite alternative meat option on the market at the moment?
So the one I'm most excited to potentially try if I ever get over to the States is work from Wild Type, who make cell cultured salmon, and their main goal is to produce a sashimi grade salmon, which looks identical to the real animal derived meat, and they've got all the perfect fat striations and the texture is perfect. And honestly, I think they're doing the most incredible job. A lot of cell cultivated meat companies mix homogeneous massive cells into plant-based meats because it is just hard to get the texture and appearance right, but Wild Type is just doing a fabulous job of producing cultivated salmon.
That's really interesting. Yeah, awesome. I never really think of salmon when I think of like the burgers. So that's really cool. So I wish I could see who posted these questions, but we'll stick with anonymous for now. So if you've seen the recent analyses by Hamburg and Vergeer, there's a question asking, what do you think of the recent analyses that predict a long horizon in terms of arrival for cellular agriculture products?
Yep, I think they're really accurate. This is getting cell cultured meat onto the market is not inevitable. There are many, many hurdles that stand in the way. One thing I would like to raise is cell cultivated meat will be more efficient and better for the environment is again not inherently built into the technology. Yes, it uses less land. Yes, less carbon is emitted, but the energy use can be a lot higher. For some products, it is higher, and unless researchers are actively figuring out ways to reduce that energy input, it's not necessarily going to be better. There's a long horizon. I want to believe that it's going to be available to us soon, but there's a good chance that won't be the case. I think it's a little bit different with precision fermentation. The technology is slightly easier, and it's better understood. It's more established. But yeah, absolutely, cultivated meat is not an inevitable destination, and it might be a way off.
Yeah, that seems like a reasonable response at this stage. Regarding your time at Nourish Ingredients, what kind of challenges are Nourish encountering with regard to scaling? Is scaling precision fermentation relatively straightforward, or are there some significant challenges there that we don't know about?
So of course, taking a small scale reaction and making it bigger is always going to encounter challenges. But again, compared to growing up mammalian cells, working with yeast and bacteria is so much easier. And that's, those products are going to come to market far before we see cell cultured meat. Yeah,
cool. That makes sense, because if it's essentially the exact same thing that we already do for products that do exist, like insulin
the iteration times of yeast and bacteria are very, very quick, and you can they multiply far more rapidly than mammalian cells.
Mhm.
And yeah, this is technology that we use to create heaps of other products. So it's not all, precision fermentation isn't all that new. Growing meat from mammalian cells in the required proportions, that's a challenge. And another thing that's perhaps important to keep in mind, people sometimes go off talking about how this is an answer to food insecurity. At the moment, producing meat via cellular agriculture is not at that level. This is not going to, it's going to be a really niche product, and it's not reaching people who are facing food insecurity challenges. Growing meat in bioreactors is not going to answer that problem right now.
Yeah, it's a great point. Cool. Okay, so on cellular agriculture again, so this is more of an animal welfare related point. How will we combat the need to return to the animal for both biopsies and serum in an economically viable way?
Yep, so on the serum question, that is being addressed pretty effectively. Companies who have cell cultured meat on the market do not use media that uses serum from animals. It's all animal free. And I don't think any product is going to hit the market that has used a serum that was from cows, because that completely defeats the purpose.
Yeah,
that's been answered. In terms of going back to animals for biopsies, I don't think animal agriculture is disappearing anytime soon, and it's actually a very important way in which we can help farmers transition out of intensive agriculture and keep them involved in the process. A lot of farmers are quite reactionary towards the idea of cultivated meat as their whole livelihood being lost, and involving them in the process and keeping smaller scale farms going, at least for a considerable amount of time, will keep them involved and ensure that we don't just lose a whole industry and people don't just lose jobs. So that's going to be important going forward, is involving the farmers in the process.
Yeah, sure. And I guess on the biopsies point as well, they're very, very micro scale biopsies that we take.
They are micro scale, and going forward, people are developing these massive cell banks, which will hopefully be distributed evenly across that's a big issue, trying to keep them private versus government regulated. So animals are here to stay for a while.
Yeah. So a question from Kyle, who has a name, which is amazing. With the first precision fermentation meat alternatives in the Australian market, so we're not talking about dairy or precision fermented egg products, be price competitive with meat? Is that a priority?
We're not using precision fermentation to make whole meat replacements. Precision fermentation produces proteins or lipids which are then added to meat replacements, but they themselves aren't producing a meat replacement. Does that answer the question?
I think it probably does, in that.
Yeah. So in terms of replacing meat like for like at a bio-identical level, cell culture techniques are required. Precision fermentation can't do that. Yeah,
that answers things. And I think this is our last question for you, Ruth. Regarding, again, collaboration within the Australian agriculture space, how much collaboration does Nourish do with other players?
Yes.
There's a lot of duplication in these early stages.
So that's a really exciting question, because recently Nourish actually partnered up with VAL, and they became the world's first precision fermentation to collaborate with a cultured meat company, which is really exciting. So Nourish has an official partnership with VAL, and that's great to see these hybrid products coming forward, because I think blending two different technologies is a really exciting answer to the alternative protein problem. So yeah. It's good amount of collaboration happening.
That's awesome. Okay, so I think we are finished up with questions for now, Ruth. Thank you so much. So with that covered, we will move on to the first of our pre-recorded talks here. So I will try and get that up on my computer, and then I'll be screen sharing from there. So I'm obviously very adept at all of these things, and that's why I was chosen for this. Does that appear to be working for people?
I can see that.
Amazing. Okay then, we will pray to the gods of buffering that things progress well. So this is a talk prepared for us by Felix Septianto, who, as I explained before, leading expert on marketing and the influence of emotions in consumption behavior. And yeah, he's got some funding to look specifically at consumer acceptance of cultured meat. So I'm really keen to take a listen. But I might just need to get his audio working first.
Sorry, Tom. It doesn't sound like it's transmitting the audio through screen share.
Ah, okay. That is a thing that we need to solve. Okay, I've got share down.
The acceptance of culture.
There we go. Can hear that fine.
Amazing. Look at me go. I always laughed at my lectures when they couldn't figure out Zoom, and I think I should apologize to each and every one of them. Okay, let Felix take it away and hopefully everything's clear.
My name is Felix from EAUQ Business School. So this evening I would like to present some information about the acceptance of cultured meat. So as we know, cultured meat has different names, also known as cell-based, clean, synthetic, or cultivated meat. It's basically produced by extracting cells from an animal by a harmless biopsy and then growing them into meat cells. This process offers a potential alternative to factory farming and may reduce associated ethical, environmental, and social issues. In particular, if it is successfully commercialized in Australia, it could reduce multiple environmental issues associated with conventional meat production. As I pinpoint here, it's 78 to 96% lower emission, 99% lower land use, and 82 to 96% lower water use. More importantly, it even eliminates ethical concerns around the treatment of animals. Despite not yet being commercially available, there has been a huge amount of interest in this new potential field, including from large investors. So in the US, for example, Tyson Foods and Cargill are the two big companies, private companies, that have invested a lot of money on cultured meat. Now, research at the moment generally finds that acceptance of cultured meat is quite high, with around 2/3 of participants, or 2 out of 3 participants. In general, 2 out of 3 people reporting willingness to try cultured meat in most samples. And in general, acceptance levels are particularly high among people who are younger, males, those from urban areas, those with higher level of educations, and those who are politically liberals. On the other hand, negative attitudes are associated with certain psychological variables. So usually the ones who have negative attitudes are those who have high in conspirational ideation. They have also food neophobia. Basically, they avoid new food, and they also have high level of meat attachment. Now, these findings are primarily from research conducted in the US and European countries. However, recent cross-cultural investigations in China and India also suggest similar results. Now, if that's the case, then it seems that cultured meat is kind of like in general is good, right? But it's not necessarily like that. So if we kind of like go back to the findings in general, so 2 out of 3 consumers, or like 2/3 of consumers, reporting willingness to try cultured meat. So there are 2 issues with this, right? So the first one, 2 out of 3 people, which means that 1 out of 3 people still reject cultured meat. And if we talk about millions and millions of people, 1/3 of those millions are still a lot of people. So we can see that there are still many people who dislike or not accept the idea of culture. So the second one, what we call in social psychology, is attitude behavior gap. So from the research, from the survey, 2/3 of participants, they report willingness to try cultured meat. Now, willingness to try cultured meat, it doesn't mean that they are actually going to buy or consume cultured meat. It's just on the survey, they report they're willing to try, which means that there's still a lot of issues associated with cultured meat. So essentially, we still need to understand the barriers of cultured meat acceptance so that we can increase the acceptance, and hopefully we can commercialize this, essentially from the business perspective. Now, what are the potential issues? So research generally finds that acceptance is quite high. However, the problem is about safety concerns, health and nutrition concerns, concern for the loss of farming jobs and traditions. But the most pervasive barrier is the perception that cultured meat is unnatural. In fact, research from Sygris and Sutterling, they found that the health risk from cultured meat was less acceptable than identical risk from conventional meat because cultured meat is perceived to be unnatural. Now, the question is then, how can we address this barrier, the barrier that cultured meat is unnatural? Now, researchers have attempted to overcome this barrier, the perception of an unnaturalness, using different communication strategies, but they have limited success. So I have 3 points that are kind of like I can share based on prior research, the main points. So the first one, they have tried to focus on the unnatural nature of other food productions. Basically, they say like, it's not just cultured meat, but other food production also somewhat unnatural. But it doesn't really help. It yields small improvements in cultured meat acceptance. The second one, in fact, that when people learn about cultured meat, they actually improve attitudes toward conventional meat. It's actually backfire. So people who are ambiguous about conventional meat, they actually, when they learn about cultured meat and see that cultured meat is unnatural, their perception about conventional meat actually becomes more positive because they would see it as more organically produced meat. So we can see that this approach is also not successful. And finally, framing of cultured meat can also influence acceptance. So when we use the word clean meat as compared to lab-grown meat, so clean meat is seen as more positive than lab-grown meat or cultured meat or synthetic meat. Or using technical or high-tech description as compared to non-technical description about cultured meat can also influence. So people actually dislike or show negative attitudes when they read a technical description about cultured meat. So this is kind of like worrying because this has been a very dominant frame in early media coverage of cultured meat. So we can understand that this is arguably inevitable because its technological advanced nature is what's made cultured meat actually newsworthy and interesting. However, from this research, it is plausible that such technical framing or technical description actually causing consumers to develop more negative attitudes. So to address this, I guess we need to kind of like go back and then see from the psychologically speaking, how do people form the perception that cultured meat is unnatural, right? And recent research suggests that when people reach conclusion about naturalness or unnaturalness about food, it is intuitive, non-analytic, and emotional process. So this form is kind of like fast and really high influenced by emotion. So from this perspective, people reach a conclusion that cultured meat is unnatural because of their gut feelings or intuitions. So it lies outside their conscious awareness and it's infused with emotions. And this might explain why acceptance strategies that I have kind of like outlined before, it might not be really successful because they are purely target analytic processing or information. And that's why they might have limited success. So this means that apart from, rather than thinking about just purely cognitive or purely providing cognitive information about cultured meat, we need to learn more about the emotional side of it. How can we target the emotional side of it to increase the acceptance of cultured meat? And among different emotions, the most relevant one, I guess in this case, is disgust. So disgust at its core is a food rejection emotion. So maybe you remember, if you know Inside Out, one of the contexts when disgust is important is because when disgust try basically reject food that the main character dislike essentially, and it has been defined as the underlying motivation of avoidance responses. In fact, there is evidence that this emotion evolved to protect us as organisms from diseases, and it's considered as a part of behavioral immune system. So it is disgust as an emotion has evolutionary functional, which is really useful. However, at the same time, disgust can also kind of like become a huge barrier to cultured meat acceptance. Now, the interesting thing is that even though previous works in this area, they have identified disgust as a barrier associated with perception of unnaturalness of cultured meat, there is little empirical evidence studying how to deal with acceptance of cultured meat from this emotion perspective. And I guess that's from my perspective, because my expert is studying the context of emotion and consumer decision making. This emotion is studying this emotion is kind of like the way we can go forward to increase the acceptance of cultured meat. And obviously, there is no empirical evidence about this yet, but these are the three potential ways that we can kind of like move forward by leveraging the emotion of disgust. So the first one, we can leverage information processing associated with disgust. So essentially, like based on prior research in social psychology, they have what we call control level, essentially how we process information at a concrete level or at an abstract level. So disgust, because disgust motivates us to kind of like reject something and then see something from far away, essentially, because we don't want to go closest. We don't want to be close to something that we discuss about, essentially. So because of that, disgust actually elicits high control level and abstract mental processing. So if that's the case, then it might be plausible that we can increase cultured meat acceptance by using an abstract information. Essentially, information that is more kind of like high level, thinking about why instead of more concrete and more detailed information, essentially, or more detailed description. The second one, we can also evoke disgust associated with conventional meat production. So although cultured meat might be disgusting for some people, we can also evoke disgust associated with conventional meat production. And this is interesting and a plausible way to move forward, because disgust is also a moral emotion. So there is a line of research in social psychology showing that when we feel disgust, we also are motivated to do something that is in line with morality, right? Because we feel disgusted also when we think of immoral behavior. So disgust is also a moral behavior or moral emotion, sorry. And because of that, it's possible that when we think of, when we evoke disgust associated with conventional meat production, it can help people to think that like, well, even though I feel disgusted about cultured meat, but that's the right thing to do. And that's why I might be willing to consume cultured meat because that's the right thing to do, essentially. So that's the second one. And the last one, we can also kind of like, you can say, mix disgust with other emotion. So this is the idea of mixed emotions, in which that mixed emotion kind of like motivates people to kind of like try something new. So when we mix disgust, let's say, with curiosity or fascination. So when we think of like, even like say arts, right? We call it grotesque art. So when we feel at the same time feel disgusted and attracted. So we can use those mixed emotions when we kind of like promote cultured meat, so that people are more likely as well to try consuming cultured meat and be more accepting of culture. So these are the three ways that at the moment we can think of when we try to build on disgust to increase acceptance of culture. So that's the end of my presentation. Thanks.
All right. Personally, I love that kind of stuff. I'm not sure what other people thought of it. Maybe you can go thumbs up, thumbs down, yes, no. But yeah, I think this is probably, as I said at the start of this event, I think moving forward, consumer acceptance of meat alternatives is one of the larger barriers that we're facing. We've heard from two scientists who are doing the work and making progress with things. And we're seeing a massive influx of investment in terms of new startups, in terms of philanthropic donations as well. And there's clearly a lot of energy and a lot of belief that the products can be made. But this consumer acceptance thing does seem important. And I liked Felix's point that I think the willingness and action, or willingness behavior gap, whatever the specific terminology was, probably seems like a very good thing to keep in mind, because most of the research that I've seen about this, or most of the statements that I've seen about this, tend to be based purely off surveys where we ask people, would you consider trying this? And they're useful, sure, but like political polls show you just how bad surveys can be. And political polls don't have the same kind of underlying emotional responses to the naturalness, to unnaturalness, to new things that we see here. So yeah, watch this space, watch Felix's space, because as I said earlier, he's got a research grant to be working on this. So yeah, there'll be more stuff coming, hopefully, from Felix in future. We'll just check if anyone did send through questions. We'll take this as discussion questions, if you like, just briefly. So Tom sent through the question, do you think framing should be different for different stakeholders, perhaps use technical terms for scientists and non-technical terms for consumers? I'm not Felix, but that definitely seems reasonable. And yeah, I think what we've done sort of here today is a pretty good example of that. You always get different communication with different groups. Here we're talking very technically about a lot of different processes underlying alternative meats, because we're already on board, and because you need to be able to communicate in that way to actually understand things. But Felix's idea of just very high level, I suppose, vague communication, that's definitely what we see brands doing normally with marketing their products. So that seems like a good way to go for public communication. If no one wants to jump in for any discussion points or comments or queries there, I think we'll head on off to Mark's talk now. So as I mentioned earlier at the start, Mark is another chemical and bioengineer who leads the biomimetic systems engineering laboratory here at EAUQ, who are hiring, which I believe he may talk about, but if not, they are hiring if you're a tissue engineer in need of a job. So we'll dive in and hear what he has to say about tissue engineering for alternative mix now. Or not, we'll make sure that we can.
Of Queensland. I was at QUT for a new senior lecturer at University of. Hey everyone, my name is Mark Allenby. I am a new senior lecturer at University of Queensland. I just started a few months ago. I'm also an adjunct at Queensland University of Technology. Before University of Queensland, I was at QUT for 3 years, and then before that, I was at Imperial College London in the UK for several years as well. I'd like to thank Effective Altruism UQ so much for inviting me today. I'd also like to thank them for allowing me to pre-record this lecture. My wife and I just recently gave birth to our first daughter, and baby girl needs my help. And so pretty much all my extracurricular activities after work have been canceled because I need to help out around the house. So I've been asked to join you today to talk about some of the engineering challenges related to growing tissue within the laboratory and growing tissue from cells. And so cellular agriculture, thanks so much for putting this on to Philippa, as well as I know Bianca Lee is also involved with Cellular Agriculture Australia. I'm creating my own laboratory, the Biomimetic Systems Engineering Laboratory. Please check out our website and let us know if you're interested in pursuing a PhD or a postdoc position with us. I'll talk more about that later. And so first I'd like to talk about the motivation for why do we care about tissue engineering? Well, a lot of my research is in the biomedical domain, not so much the agricultural domain. So we care about tissue engineering to provide implants for patients in the hospital or drug disease models in the laboratory. And so in terms of implantation, globally, over 2.5 million people are on wait lists for life-saving organ transplants. And annually, over 40 million tissues are transplanted. And you should be including blood as a tissue because blood is a tissue. That number actually goes to 500 million. However, donation of tissue is the only way to really source most of this transplantation. And so currently, transplantation completely requires altruistic people to donate their blood, to donate their tissue, to donate their organs at the hospital to save other patients' lives. This can be really difficult if you have a particularly rare condition, if you require a tissue urgently or regionally. And you can see in the US this difference between the number of people on the organ wait list and the number of organ transplants and donors that exist per year. So according to the US National Institute of Health, tissue engineering is the combination of scaffold cells and bioactive molecules into functional tissue. And so they say that there are 3 components, and we'll talk about them today, right? So it's scaffolds, which is like a construction scaffolding, right? A material scaffolding which supports cells, where cells live, and cells start to populate it into your building, into your tissue over time. You also require the cells themselves and biochemical molecules that influence cells to grow. The goal of tissue engineering is to assemble functional constructs that rebuild, maintain, or improve damaged tissue or whole organs, right? To kind of introduce some of the engineering challenges that might exist, I'm going to talk about an example implant and some of the steps that are typically associated with tissue engineering. So let's say we have a patient in the hospital who has a damaged meniscus, cartilage within his kneecap and his leg, right? And so this could either be diseased or this could be damaged. The goal of tissue engineering is to assemble functional constructs that rebuild, maintain, or improve damaged tissue or whole organs. The goal of tissue engineering is to assemble functional constructs that rebuild, maintain, or improve damaged tissue or whole organs. what is typically done in the tissue engineering cycle is stem cells need to be isolated from the patient, one area of which could be stem cells being isolated from the bone marrow. The bone marrow is the spongy inner part of legs, or spongy inner part of bones throughout the body. Spongy inner part of any bone throughout the body has marrow where there exists marrow stem cells. Now, the number of stem cells that you might get from a biopsy is very small, and the number of stem cells you might require for a functional transplant is very large. And so you'd have to expand this number of cells into very high quantities. But stem cells are very fragile, so it's difficult to grow them in very careful culture conditions to allow them to survive and allow them not to be affected over time. And so being able to have this scalability and control over culture processes is a big engineering challenge. Once you have enough cells or stem cells, you would then think about seeding these cells into your scaffolding, into your material. And you'd want to design material in such a way that it might mimic the shape, the structure, the mechanics, or the biochemistry of the patient's natural tissue. And you would derive these kinds of parameters through medical imaging, chemical formulation, or different manufacturing methods. Once you have cells sitting inside of your scaffolding, you'd want to continue to grow these cells so that they would bridge all the gaps, all the pores in your scaffolding and form fully developed tissue, or something more resembling fully developed tissue. There's a lot of challenges here as well. I know my picture is covering some of these images, but when you create volumetric tissue, it's difficult to transport nutrients and metabolites or growth factors throughout your construct. It can be difficult to stimulate the mechanics that might be essential for that tissue to mature. So let's say if you have meniscus, you might want to apply different types of pressures or stretchings of that meniscus so that it forms different kinds of proteins or extracellular matrix so that it has the mechanical properties to make it durable and strong once it's implanted. And also this needs to be a process that's well automated and well controlled. All of these are manufacturing and engineering demands. Once you have your finalized tissue, you need to prepare it, and it needs to be prepared in such a way that it could be stored for periods of time, that it could be shipped to different parts of the world, and a surgeon could easily prepare it for implantation into a patient at the hospital. And so all of these are different kinds of life cycle concerns from an engineer standpoint when it comes to growing tissue in the laboratory for a clinical dimension. And why do we care about tissue engineering implants? Well, it's because current implants are either autologous, which are derived from the same patient. It depends if that patient has healthy tissue to be able to provide for this replacement. Allogeneic, from a separate person that is freely giving a donation of their tissue to this patient. So there needs to be that person who's being so altruistic. There's always a cause for person versus person graft rejection based on non-matched blood types or potential diseases. Synthetic grafts can also be problematic. They don't necessarily have the correct biocompatibility or mechanics to match that patient tissue. And so tissue engineering is aiming to solve these clinical shortcomings. And while growing lab-grown tissue for implants is in its infancy, there are some initial successes. So there are 20 clinically approved, FDA-approved implants, such as mucosa grafts, cartilage grafts, skin grafts, ocular grafts, cardiac sheets, not larger pieces of a heart. And these are all grafts that are simple tissues, simple flat sheets of tissue. These aren't large volumetric constructs or organs that have several functions and several tissue types, cell types. No, these are very simple tissue. And so there's a lot of room for tissue engineering to grow to become more complex and tackle these more difficult challenges. This is a relatively large market for being emerging, but it's also the most rapidly growing segment of the biotechnology field. That includes agriculture, gene editing. So tissue manufacturing and regenerative manufacturing is set to almost triple in value within 8 years. And there's a lot of federal funding for different national research institutes in other countries, a little bit less in Australia. And so I hope that Australia comes to its senses. Tissue engineering is also more than clinical implants. And so I was saying earlier, if you're growing human tissue in the laboratory, you could use that human tissue to test the efficacy of drugs. Instead of using animals in preclinical testing, you could use lab-grown human tissue that didn't have to kill animals to produce it. like we're talking about today, this lab-grown tissue could be derived from animal cell sources, a small animal biopsy that's grown to become an animal tissue in the laboratory that could become meat or different agricultural products like leather. Finally, if we find good methods to grow cells at high densities, at tissue densities, this would also be very valuable to other markets, such as the production of monoclonal antibodies for vaccines. Monoclonal antibodies are derived from cell culture. If we could grow cells at very high density as tissue, we would achieve better manufacturing efficiencies for pharmaceuticals. And so the problems here is that lab-grown tissues remain expensive, non-physiological, and inconsistent. And so my laboratory aims to optimize tissue manufacturing. And to kind of go through some of our technologies, we perform the experiments in the laboratory, isolating cells from patients, growing them up. We also develop imaging algorithms to be able to image the quality and the function of our tissue, but derive those images and those videos to produce data sets that we can feed into mathematical and computational models to predict better cell culture experiments to more efficiently or more exactly, more precisely manufacture tissue. And this is to engineer better biomanufacturing and medical manufacturing processes. So I'll give a few examples of a few different research projects I've been working on very quickly. I've been talking about how essential blood is as a transfusion product. Blood is produced within the human bone marrow, the spongy inside of bones throughout the body. There exist blood stem cells, and these blood stem cells produce all the other types of cells in your body. But it's really difficult to mimic the human bone marrow. There's so many different types of cells. There's structural stromal cells, bone cells, blood cells. There's also vessels that control the content of nutrients and metabolites throughout the marrow. It's difficult to get this right in a laboratory context. It's very complicated. What we did is we produced a bioreactor, right? And this bioreactor was comprised as a spongy scaffolding where stem cells could exist, right? Just like in the marrow. And then there were hollow fiber channels that acted as vasculature. And within these hollow fiber channels, we perfused medium such that we could replenish the nutrients and metabolites of our cell culture, but also our hollow fibers were engineered such that they would allow the passage of red blood cells continuously. So we could continuously harvest the red blood cells that were produced within our bone marrow mimicry in the laboratory, right? And so these are some blood cells that we were isolating. We wanted to optimize this device, so we began performing image analyses of 23 different types of cells throughout our device and feeding that into mathematical models to understand more optimal distributions of growth factors, of nutrients, of metabolites, so that we could perfect our bioreactor operation, and we further boosted our red blood cell manufacturing efficiencies 50-fold. We're also very interested in 3D printing. And so with one of my PhD students, Trent Brooks-Richards, and a great collaboration with Mia Woodruff over at Queensland University of Technology, we looked at 3D printing of very fine fibers and very fine pore sizes using a technique called melt electrowetting. And using cell culture and image analyses, we very finely measured the kinetics of cell regeneration in different pore sizes throughout our constructs and understood how these different pore sizes would affect our resultant tissue alignments. Using mathematical modeling, we fed this imaging data into mathematical models to understand single cell regeneration kinetics, such as diffusion or migration of cells and proliferation of cells. And of course, we're looking at scaling this up further to look at differentiation. And the importance here is that if you have a given tissue where you want a specific type of proliferation, regeneration kinetic, morphology, you could say you should use this kind of scaffold geometry to be able to direct your cellular growth for your desired tissue output. And so we performed cell culture experiments, ran imaging analyses, performed mathematical models that would then feed into much better tissue production into the future. Is looking at mechanically complex tissues as well. And so one tissue we're interested in is the popliteal artery. The popliteal artery is on the back side of your leg. And this is an artery that undergoes a lot of different contortions every day when you walk, when you run, when you crouch. And coincidentally, not necessarily coincidentally, people are finding correlations between this high mechanical activity and the fact that disease so commonly manifests in the popliteal artery. So 80% of peripheral artery disease specifically manifests in the popliteal artery. And so we looked at how this popliteal artery stretches in different orientations. We began performing histology to look at the muscle content of this artery in different locations. And we began designing different cell culture platforms that could replicate some of the different mechanics that goes on in this artery, as well as the radial distension of this artery. So this is multi-axial mechanical simulator for cell culture. And we found when we seeded cells on these constructs, we're able to control the orientation of cells with different mechanics. And we could also augment the regeneration kinetics of these cells. And not only that, we also influenced mesenchymal stem cells to differentiate into some early smooth muscle cell phenotypes just with mechanical stimulation alone. And so again, this is a project where we performed imaging analyses. These imaging analyses fed into simulations where then we built cell culture platforms to better create synthetic tissue. Another project that's a little more in its early stages is a project about intracranial aneurysms, which are bulging vessels throughout the brain. We created software that could take in just everyday hospital images, and we're up to a repository of about 100 patients and about 150 images so far, where we're running an imaging software that we wrote, that we developed, to be able to automatically identify the location and the appearance of intracranial aneurysms as a second set of eyes for the untrained radiologist. This can even pick up aneurysms in very complex cases. And now that we have the ability to detect aneurysms, I'm sorry my picture is covering this up, we're also performing simulations, both numerically and 3D printing experimental simulations of blood flow to understand if we have these aneurysms, which aneurysms have the different fluid shear stresses, which could mean that they're prone to rupture in the future. And so again, this is a project we're performing imaging analyses, we're feeding this into mathematical models, and we're producing experimental testing systems. And these are systems that we hope to inoculate with cells and create bioengineered aneurysm testing systems as well. And so just to kind of summarize, these are processes that we're looking to engineer better biomanufacturing and medical manufacturing processes. And so, of course, I'd like to thank my team. My team performed all this research, the most part of this research, at Queensland University of Technology because I've just arrived at University of Queensland. A number of students are currently with me at University of Queensland, Sabrina and Mitchell. The results today also came from Trent and Brenna, who are doing amazing work that I'm collaborating with at QUT, and Cody and Victoria, who finished up their degrees successfully and were currently publishing their results. Lloyd and Yogi Sen. So thank you for listening to me. I'd like to thank our funding bodies and our industry partners. Specifically, I'd also like to thank Maria Woodruff, who I was in her laboratory at Queensland University of Technology and derived most of the data that I'm presenting today. I'd like to thank our clinical collaborators, Craig Winter and Isharan Lang from Royal Brisbane and Women's Hospital, Thomas Lloyd and Yogi Sen Sivakumaran from Princess Alexandra Hospital, and our great mathematical modeling collaborators and international collaborators. So please reach out to me if you're interested in engineering more realistic tissue growth platforms, if you're interested in performing and creating software around image analysis or analyzing your microscopy or medical imaging data, or if you're interested in developing computational simulations of mechanics or other features within physiological systems. Also reach out if you're interested in PhD study or other research positions. Our laboratory is live online, although it's not quite finished. Please feel free to check it out. Thanks so much.
Sweet. Awesome, we're back. Okay, So that wraps up the talks from the qualified people today. And although he's not here, I'd like to thank Mark and also to thank Felix for their contributions today. I found both those things really interesting, and I think it's particularly fascinating to see how alternative meat and the tissue engineering that we're looking to do here does intersect with the medical stuff that Mark was talking about a bit more, in that a lot of those same issues around scaffolding occur with clean meat production as well, where we can just get cells to proliferate and create hunks of meat cell, but getting that to actually do so at scale and into the shapes that we want is particularly difficult. So I am happy to conclude with some thoughts of my own or pass over to Philippa. But yeah, that's the end of our proper talks now.